All right, so today we have Axel Ahmed. He is a postdoctoral scholar at the University of California in Irvine and is supervised by Professor King Ni. He is currently interested in modeling and analyzing the role of cell-cell communication and its effects on high-dimensional sequencing data, particularly in processes such as wound healing and development. Today, he is going to present FlowSig, which is a method that infers communication-driven intercellular flows from scRNA-seq or ST data using graphical causal modeling and conditional independence. And he is going to demonstrate FlowSig's utility by applying it to various scRNA-seqs and spatial transcriptional studies. With that, hand it over to Axel.
So let me share my screen. Yeah. All right. So thank you all for coming to this talk. Thank you to Hector and Ahmed for the invitation. As Hector said, I'm a postdoc at UC Irvine, working with Ching Ni on things related to cell-cell communication and our ability to infer it from high-dimensional data like single-cell and spatial transcriptomics. For the sake of this talk, I will only focus on the aspects of this work that Hector mentioned of FlowSig that apply to spatial data to keep in line with the theme of this seminar. But the idea is like what I will say about spatial transcriptomics can also be applied to non-spatial data analysis with certain caveats. And so with that, oops. So I wanted to start with a simple statement that we all know to be true, which is that no cell is an island, despite the advent of and the advances in single-cell data, which allow us to analyze transcriptional states at this like near unprecedented resolution and look at gene transcription, which is sort of like this working memory of cells, and allows us to classify different cell types and cell states. The reality is, though, that cells don't really act in isolation. They're constantly in communication with each other. They're constantly in this dialogue between each other, both within, say, same cell types and also between different cell types. And the way this is facilitated is through what we call cell-cell communication. And for the sake of this talk, I'm going to talk about cell-cell communication through protein-based ligand-receptor binding. So what you have is that there are certain cells can secrete certain molecules called ligands. These are proteins that can diffuse through extracellular space and bind to other proteins called receptors that exist in other cells. In this way, I kind of think of it, considering the World Series just happened, ligands are kind of like baseballs and receptors are like globes. And the idea is that certain ligands can only bind to certain receptors. And so this allows certain information, certain messages to be sent back and forth between cells in this way. And this is what enables cells to really transmit this information over time and over space. And one of the important aspects of communication is that actually it can be causally driven. By that, I mean, you can have cells that secrete ligands. In this case, this blue ligand, it diffuses, it binds to this orange receptor in this orange cell. And what actually can happen is that it may trigger some sort of downstream response. By that, I mean, it triggers some sort of signaling cascade, phosphorylation, and it activates the transcription and translation of certain transcription factors that then bind to enhancer promoter regions. That then can actually, in turn, activate the secretion of other signals. And so in doing so, by receiving and sending these signals, you're generating this flow of intercellular information. And one of the questions myself, Ching, have been asking, my supervisor have been interested in is, how do we infer when these flows occur, these flows of intercellular information due to cell-cell communication? And how do we infer those from data which is not necessarily the most amenable to infer such intercellular flows, like spatial transcriptomics data? I want to talk about what I mean by that in a moment. But first, to kind of take another step back, my lab in general is focusing a lot on understanding how to build methods that make the most of single cell and spatial transcriptomics data. In particular, single cell and spatial transcriptomics that can capture whole genome gene expression. By that, so we're talking about like 10x, StereoSeq, Visium, like stuff like that, where you can profile the effect of the entire genome. And because I'm a mathematician, I tend to think about the technology and the biology in a more abstracted way. in that effectively what you'll end up with is some sort of matrix of zeros and integer numbers, where an n by g matrix, where n is a number of cells or spots in the case of spatial transcriptomics, and G is a number of genes. And the key thing here is that the number of genes is on the order of about 10,000. You typically capture between 10,000 to 20,000 genes. And why this is interesting to us is that those genes often contain information about important signal ligands and important signal receptors that bind together. And so we're able to use this to say, well, we can then, based on known ligand-receptor interaction pairs, we can profile for the presence of hundreds or thousands of ligand-receptor interactions simultaneously and in a very systematic fashion. And we can do this either from non-spatial data, or we could do this even in for spatial data, where while you don't have single cell resolution a lot of the time, you will have, more importantly, you have spatial information, which is a very important factor in cell-cell communication, in that cells usually have to be close enough to interact within a reasonable degree. And so to kind of give context, like when I started looking at single cell, so I started my postdoc in 2020. I'd come from a biomechanics sort of background, my PhD. One of the things that I got interested in with Ching and our group was looking at cell-cell communication. How do we use this high-dimensional but static data to infer cell-cell communication? And we were lucky enough to be able to write a brief review about it in 2021 in Current Opinions in Systems Biology. At the time, myself and a couple of other former postdocs, so Chin and Jishen, when we were going through the literature, there were only about 19 methods to infer cell-cell communication from either non-spatial transcriptomics, or single-cell transcriptomics, or spatial transcriptomics data. You fast forward to this year, there's this beautiful review by Eric Armengol from Nathan Lewis's lab in UCSD, and there are about 105 methods now to infer cell-cell communication. And so it is like, over the past 3 years, like single cell and spatial technologies, the methods to infer cell-cell communication have really exploded over time, and they continue to still be developed. And to give an overview of how cell-cell communication works, they work slightly different for non-spatial and spatial data. For spatial non-spatial inference, sorry, for example, using a method like CellChat, which is developed by former colleagues Suoqi and Jin, you take some single-cell RNA sequencing, which is this matrix of gene expression values, and you have some sort of labels to aggregate cells to reduce noise in measurements. And so the idea here is like you are inferring ligand-receptor interactions between different cell types. Here are the different colors of the nodes and the widths, and you infer these networks of interactions. In fact, there'll be one per ligand-receptor interaction that is present, you believe to be significantly present within the data. So you get hundreds of these networks, these multiplex networks, effectively. These are mostly to do with interactions between cell types. They do not have information about, because single cell information doesn't obviously have information about space, there is a chance that you will infer what is effectively a false positive interaction. You're usually inferring interaction based on the presence of one cell type expressing a ligand, another cell type expressing a receptor, and you believe that significant expression of those constitutes the possibility of an interaction, which is not always true. In the case that like cells may not actually be close enough to be communicating, or that might just be a coincidence. And so this is where using spatial data is really powerful for cell-cell communication, because we actually can use plot the spatial distribution of ligands and receptors. We can infer which interactions are present to with much more accuracy. The key difference here, though, is that usually with spatial transcriptomics technologies, for example, with 10x Vision, the most popular, you're losing single cell information. And therefore, it is a lot harder to say something about, say, the role of specific cell types. Even though there's a lot of work that goes into this, it's just like you have a more coarse-grained data. But what's nice about spatial data is that using methods like COMMA, which my colleague Jishen Tang, who's now at North Carolina State, developed, you can infer, based on ligand expression, receptor expression, you can distinguish between not only the presence of interactions, but you can also infer where a signal was sent and where a signal was received based on ligand receptor distributions and using methods like optimal transport. This is a really powerful method aspect of spatial transcriptomics, in that you effectively are accounting for the role of diffusion of these signals and binding and stuff like that. And I'll come back and why this is important later on and how it informed our development of FlowSig. So these are kind of usually what the sort of methods that can be inference that can be done right now. What's important about what's a key caveat though of these methods is you infer these many hundreds of interactions from these gene expression data. And the key assumption usually that is not really explicitly said often, which is that these interactions are usually inferred in a one-by-one cases. They're inferred as, and so you're implicitly assuming that they occur independently of each other. For example, if I'm inferring Wnt signaling, I'm assuming that is occurring separately of BMP signaling. However, we know that this isn't always true. We know that there are cases where, for example, Wnt may drive BMP signaling, for example, in the colon. It's very well known that those two interact with each other to drive colon cancer and maintain homeostasis. So what would be nice is if we could infer, based on the cell-cell communication inference we do, can we go a step further and ask which of these interactions depend on each other? In other words, does it say sending of Wnt cause or drive the sending of BMP in our data that we're analyzing? That in itself is quite a hard question to ask because you're asking about information between two different cells, intercellular relationships. But what you can do is effectively reverse this around and kind of think of the inverse, which is to ask, actually, if I'm receiving Wnt signal, does this cause me to then send BMP signaling, for example? So where the idea for FlowSig is that we're going to test the effect of receiving a signal on sending a signal. And by doing this, you're then kind of framing the question in terms of what a single cell or a single spot does. And therefore, we can use methods developed in the causal inference and causal discovery field to try and answer this question about these, to infer these dependent interactions. And so with this in mind, we developed this method FlowSig. It's just been published in Nature Methods to infer what we're calling communication-driven intercellular flows. It's a bit of a mouthful, but effectively what we mean is that given that you have your gene expression data with spots or cells and 10 to 20,000 genes and some cell-cell communication output, which FlowSig can either infer for you or you can provide, we can take this input and spit out what is a systems level network of dependent intercellular flows. By that, I mean a network that describes when you receive an inflow of one signal, which other signals does that drive the occurrence of? For example, if you receive Wnt, does that cause you to then drive an outflow of BMP, sonic hedgehog, or something like that? And the idea is that we're going to infer what I call a systems level network. It infers all possible interactions that are going on in the tissue based on what you already see in the data. And this is not based on any prior knowledge necessarily. This is based on what we directly observe in the data. And there are three sorts of levels of network of nodes in the network that I'm going to talk about that are key to making FlowSig work. And effectively, it all revolves around the notion that sometimes when a cell receives a signal, it will then send another signal back out. And the idea, as I won't talk about the non-spatial, but it works. Effectively, the pipeline is the same for both non-spatial and spatial data analysis. For the sake of this seminar, I'll only talk about spatial data analysis and the application that we use in the paper. So there are three main steps to FlowSig, all equally important in some way. One is variable construction. So how do we take the gene expression, cell-cell communication, and define cell wave measurements that enable us to infer these intercellular flows? Two, how do we then infer the actual network of dependent interactions? And then three, how do we kind of make sure that what we infer makes biological and statistical sense? So the first step we're constructing is to construct three different types of variables. And this is really the key because one of the fundamental issues we had with single cell and spatial data for a while was like, you do not directly know how much of it, what signals each cell is receiving or sending. You know what signal ligands, for example, are being expressed by the cell, but you didn't know to who was receiving or to whom the cell was sending these signals. And so to break it down, we had to kind of think about what is going on for like an individual cell or an individual spot. And so that led us to define three types of variables. First is what we're calling signal inflow, which is really just measuring the amount of signal received due to, say, a specific ligand. So in this case example I'm showing, this is Wnt5a and Wnt5a inflow. And the aspect here is that Wnt5a can bind to a number of different receptors, Frizzled1, Frizzled2, NCAM, And so there are many different ways that Win5A can be received by different cells in different spots. And the nice thing about using an optimal transport method like the one Jashan made comment is that you can effectively infer what is the amount of received what is the effect of received Win5A interactions based on these different receptors. And so if you have this information about the different interactions, you can simply take the sum, and that will define at each spot the total amount of Win5A received from nearby areas or the same area. The key point here is that we actually, when you're expressing ligand gene expression doesn't correlate to the amount of received ligand. And so that's why we have to kind of do this extra step and use something like an optimal transport method or some sort of like method to account for the diffusion of signals into spaces and their reception into nearby areas. The second is what we're calling intracellular modules, and these are meant to be proxies for the gene regulatory processes that process the signals and then convert them into other signals. And the way we're going to do it is we're going to, rather than say directly take individual transcription factors, which increases the number of variables and makes the problem computationally intractable, we're going to take a coarse-grained approach, which is we're going to perform effectively dimensionality reduction. And the way we're doing it for this is effectively using matrix factorization. So the most common would be non-negative matrix factorization. For spatial data, we use a method called non-negative spatial factorization, which was come up with by Will Towns and Barbara Engelhardt, where you effectively do matrix factorization, which is to say you decompose your gene expression or spatial expression matrix into K modules, where K, the idea is that K, the number of modules is significantly smaller than the number of genes. So we're talking about the order of 10 to 20 compared to like, say, 10,000 genes. And you get these two different matrices, which represent one, what are the genes that comprise these modules, and most significantly, where the idea is like within each module, you have genes that are significantly correlated with each other within the module, more so than, say, genes between modules. And then you also have this other matrix, this N by K matrix, which it indicates module enrichment or membership. So how strongly are spots expressing that certain module? And the idea here is that we're hoping two things. One is that these modules capture significant information about different cell states. So we're adding a little bit of cell state or cell type specificity into our regular network. And two, that they also, we believe that they capture important information about the downstream transcription factors that are really what are the indication that certain signals have been received. And they also what drive gene regulation. And the reason we use NSF is that we believe that we're hoping that we'll also capture something about spatial information. The idea is that these modules here are much more, not only do they vary, are they related in terms of gene expression, they're also spatially correlated. So we're trying to get these spatially coherent models to model sort of to account for the fact that not all signals are received in the same way across every part of the tissue. And so you get these sort of localized spatially coherent modules using this NSF method. But you could effectively use any other dimensionality reduction method. In this case, we use NSF, but you could use any other sort of method or spatial clustering method that you'd like. The key thing here is what I'm showing is that this is a soft assignment of module enrichment rather than, say, a hard assignment in the way that cluster assignment is. And then finally, the easiest variable to define is signal outflow, which is really just what signal ligands are you expressing? What signals are you sending out? In this case, this is the simplest one in that we literally just take gene expression. The other two are taking gene expression or cell-cell communication inference and mapping it into a new variable. This is one-to-one gene expression of signal ligands. And we're only considering ligands that are, say, spatially variable and have some sort of meaningful autocorrelation structure. And so and the idea is like these three variables are connected through a very simple notion, which is based on the fact that cells have to receive signals, process them before they then send them out again. And so we believe that the receiving of signal inflow then drives the intracellular processing via these module enrichments, which then drives an outflow of signals. So we have this very simple sort of like we assume there's this very clear directed hierarchy of information in the cell. Inflow, modules, outflow, receive, process, send, based on this very simple idea of how cells receive and send signals. And so what do we do with these variables? How do we learn then where the directed dependencies are? There's two steps of this, and they're based on this field of causal structural learning. I'm calling it causal structure learning, not because I think FlowSig actually really does anything causal. I don't think it's at that point yet, but that's the name of this field for those if you're interested in looking in the literature. It is called causal structure learning or causal discovery, and it requires two things. But effectively, it revolves around the idea that given you have some data, can you infer where the directed dependencies that describe causal relations directly from this data, often without prior knowledge? And it usually requires two steps, or it does for FlowSig. The first is a statistical method called conditional independence testing, which says which variables are likely to be statistically dependent. In this case, which are dependent based on partial correlation, although you could use other non-parametric tests, but they will take longer computationally. And so this, in terms of the network, this gives you where the undirected edges might be. Where might an edge be connected between signal inflow modules and signal outflow variables? And we use conditional independence because if you were to say do pairwise independence, just mere pairwise correlation, you would actually over-infer the number of edges and number of dependencies that exist that are present that drive statistical relations in dependence relations in your data, whereas conditional independence gives you sort of speak the right amount of edges. And the way to think about conditional independence is that you have two variables A and B. If you then know information about a third variable C, knowing information about that third variable suddenly makes A and B, the first two variables, independent. A very famous example of conditional independence is the fact that the future, in terms of time, the future depends on the past. But if you condition on the present, then the past and the future become independent of each other because knowing the present gives you information about both. And so you develop, and so that correlates to a very clear causal structure: past, present, future. And so that's effectively what we're using to work out where does the initial network structure look like. But the key caveat of conditional independence is that while you understand which variables might be significantly related, you don't get a clear idea of where the directed dependencies are. By that, I mean, it has been shown and it can be proven that you cannot recover a unique graph of dependent causal relations directly from statistical conditional independence relations. In fact, actually, for the set of conditional independence relations, you will get many graphs. And the more nodes you have in your graph, the greater the number of graphs that there could possibly be. And so a lot of the work in causal structure learning is figuring out algorithms to kind of figure out what is the right graph or the unique graph or the closest graph that captures the conditional independence in the most coherent or reasonable way. What is the correct orientation of edges? How do we direct them? And it's based on a lot of the work of Judea Pearl with the causal graph modeling, do calculus. And there's a really nice algorithm that we're going to use that FlowSig uses, which is called the GSP algorithm and UTI-GSP algorithm for non-spatial data. And what that stands for is GSP algorithm is a greedy sparseness permutation algorithm, which means it gives us a way of searching for the right graphs in a very efficient way. By that is rather than searching for entire edge sets, you're searching between permutation of nodes that encode certain directed graph structures. And then we're going to score them, each of these possible graphs that correspond to these conditional independence relations based on sparsity of the graph. So you will get basically graphs with different edge sets and different number of edges that can correspond to the same conditional independence relation. And we're taking the sparsest because that's been shown that's a minimal graph that will give you the right dependence structure. And it also based on the assumption that biological networks tend to be sparse, so this made sense. And so basically there's this algorithm that gives us a directed graph in the end. And so the final step is so to recap, we take gene expression cells of communication, you learn some sort of network that describes what we believe are the intercellular flows going on, the dependent signal relationships. We would like an internal checking mechanism to know that what we've found makes sense. And there's two ways we do about it. One is just simple bootstrapping, which is because the data is noisy, we're just going to resample it over and over again, run the network learning algorithm, which generates an adjacency structure, or effectively a matrix of zeros and ones. And then we're going to take the average network, which is we're going to average the adjacency matrices. So in that case, you get every edge or possible edge that could be inferred, you're assigning a score from 0 to 1 of the likelihood of it being present from the data. And so we can use this to basically only keep what we're calling high confidence edges, edges that appear multiple, most commonly during the bootstrapping. That is like the simplest way, and that is often what is done in the field to kind of make sure what you're learning makes sense. The second method is what is probably most important to us, which is that despite the fact that these algorithms are all based on trying to learn a directed graph of dependent relations from the data, they actually don't give, even those algorithms do not give unique answers. What they give is something called an essential graph, which is, or in more specific technical terms, a Markov equivalence class. These are graphs where they all have the same undirected edges, and they also all have the same what's called V structures, which is to say directed edges, if you have three nodes, X, Y, and Z, they look like this. There are two edges that point towards the middle node. Those are the only directed structures that you can't change, but everything else you can actually reorient and you would have the same statistical dependence relationships. You can prove it's been proven since the 90s. And so for us, actually, what is key then is to take the undirected edges and reorient them in the way that, well, we know that signal outflow drives intracellular processing, which then drives signal outflow. So in this example here, we're looking at the middle partition. The two vertical edges are pointing from intracellular module between intracellular module nodes and signal outflow nodes. So we know actually based on our assumption, we should orient these so that they point from modules to signal outflows rather than the other way around. And in the same way, you can't have modules affecting what you're receiving, so to speak. It can affect how you process that received signal, but it doesn't directly affect what you're receiving in that sense, in that direct sense. And we can also use this in case of there are false positives inferred, this like sort of rubric of signal inflow to modules to outflow to remove any directed edges. This very rarely happens, but it is like a sanity check. So to recap, we have three FlowSig takes gene expression, cell-cell communication that you can either provide or FlowSig will generate for you, and gives you this directed multipartite network describing which signals, if you receive the inflow of, will cause an outflow of other signals. And you have these three variables that are then that we construct to measure in each individual cell or spot, so that we can then perform conditional independence and graphical model learning to give you this network with some sort of pseudo validation. I'm calling it pseudo in that it's based on the internal data structure rather than, say, us validating it. Although we did validate this method using experimental data, which I won't talk about for the sake of time. And so how does this all kind of come together to enable us to learn something about spatial data? Given this method that we can produce these networks, what do these networks tell us? And for this, in this paper and for this talk, we looked at one example, which was really nice spatial data generated by scRNA-seq from China of this mouse embryogenesis in this in this paper by Chen and colleagues in Cell. And effectively, it gives you this whole genome coverage at near cell single cell resolution of spatial transcriptomics, and it's pre-annotated. So I think everyone in my lab has analyzed this data at some point. And so we looked at one classic example. One of the stages, the simple stage at E9.5, so early embryogenesis. We're looking at developmental mouse. We know there's a lot of cell-cell communication going on. Let's see if we can learn something new about it, or learn something from this data using FlowSig. And the key thing here is like using this data, we're able to, FlowSig will construct what are these spatial modules. Here we're constructing 20 spatial modules that describe effectively correlate and so on with the annotated anatomical structure, but also we're hoping about some information about gene regulatory state. And here I'm showing only the hard assignment. So each spot has been labeled with the module that it is most highly enriched for, just to show kind of like the spatial coherence of these different modules. But we actually will use the soft assignments for dependence lining. And so the idea is like we have the spatial data, we constructed these modules, we infer cell-cell communication using COMOT. What can we say about what's going on? And actually what I will say about FlowSig, because in this case there was you get hundreds of interactions. I think you get about 245 ligand-receptor interactions. And then from those, you whittled down to the number of dependent interactions. Even if you took that network, which is quite coarse-grained and quite sparse, there's a lot going on because we're trying to capture all possible relations that go on in the spatial tissue. And so it can be quite complicated to look at these networks, even if we're breaking it down into inflow, module, and outflows. So what we decided to do for this case is to look at a specific signal ligand. In this case of sonic hedgehog, because we knew in the past that like sonic hedgehog is very important in the context of development. It's quite significantly expressed in this data. It's also participating in cell-cell communication. So can we tease out, so to speak, the functional roles of that are going on with sonic hedgehog from this data directly? And we kind of looked at it in two ways. The first way was, given that we're seeing spatial expression and highly spatial variable expression of sonic hedgehog, can we infer which signals might be driving the outflow of sonic hedgehog? And so what we did was we take the entire network that FlowSig learns, which describes all possible interactions. You look at the node describing corresponding to sonic hedgehog outflow, and you basically backtrack through the networks. So from this outflow node, what are the modules that are connected to it? And those basically describe what we believe are the possible gene regulatory mechanisms that affect this, that drive the expression of this SHH ligand. And then you go back one step further and you ask, okay, from these modules, which are the signal inflow nodes that then drive these module nodes? And so from here, we pulled out 8 different signals that might actually be influencing the outflow of sonic hedgehog. And these are some that make sense. There are BMP, FGF, IGF, insulin growth factor, Wnt5a, all important morphogens that you see in development. And interestingly, we also see that actually that inflow of sonic hedgehog seems to also drive an outflow of sonic hedgehog, which suggests some sort of like maybe self-activating pattern. And it's only regulated through 4 spatially coherent modules, 2, 5, 11, and 14, which I'll talk about more in a bit. And so this gives us a way of classifying what we're calling the upstream drivers of sonic hedgehog. We can also kind of do the converse, which is to say, okay, we know sonic hedgehog is diffusing and being bound in the data set. It is being diffused, and it actually has pretty significant communication activity in the tissue. Which signals and which spots, given that they're receiving sonic hedgehog, are responding in turn? And as you can see here from this FlowSig network, we do the converse. You take sonic hedgehog, you look at the network for which modules are downstream of this inflow node, and then which outflow nodes are downstream of these modules. And so we can infer which other signal ligands may be dependent on the receiving of sonic hedgehog. As you can see, there's quite a number of different ligands that FlowSig infers to be dependent on receiving sonic hedgehog in some way. That is not to say they are only dependent on sonic hedgehog, but it does suggest that sonic hedgehog has a pretty important role in the way it regulates the presence of other signals in this embryonic data set. And so this is effectively just looking at the network itself that FlowSig produces. We would like to do a little more. We would like to ask ourselves, okay, we believe we can show that sonic hedgehog drives the presence of, say, Wnt or BMP, but how? And by that I mean through what transcription factors, what downstream regulatory genes are these intercellular flow mechanisms acting on? Whenever I talk to biologists, the question is always like, what genes can you give me so that we can go and test these in experiments? And so so that's kind of the reason that also motivated us to include these module nodes, because they contain a lot of this rich information about gene regulatory mechanisms. To give an example, we'll first look at what happens with upstream drivers of sonic hedgehog. So what other regulatory transcription factors that will be driving the expression of sonic hedgehog in this data set? And so the way we go about this is we look at this network, we look at there's 4 sonic hedgehog outflow depends on 4 modules. And from these modules, we can extract what are the top contributing transcription factors that drive basically the expression or enrichment of these modules. So in here, we took the top 10 module specific transcription factors. So these are genes defined by their membership scores to these modules. You take the top 10 with 10 transcription factors with the highest scores. And then what we do is we fit random forest models. So we effectively say we're asking which transcription factors have their expression that is most predictive of sonic hedgehog expression. And The reason we're using random forest models is that it effectively gives us a way of ranking the predictive power of different nodes and different transcription factors based on what's called the Gini importance. So the idea is like if you took away that node, how much would your prediction of Sonic Hedgehog outflow change? And so effectively we can rank the importance of different upstream transcription factors in terms of their regular or predictive ability power of Sonic Hedgehog outflow. And so we looked at, and when you look at the top five, you can see that there's a number of fork-head box transcription factors. There's MYC, there's a zinc finger transcription factor here. And what's nice about this is that the FOXA1, FOXA2, FOXP2 transcription factors, these are all known in the literature, so it at least confirms that we're on the right track and that FlowSig does, using FlowSig, we can infer like these are the key upstream drivers of sonic hedgehog signaling in this data at this time point. The other two have not been previously reported, so they could be new, or they could also be false positives. What's interesting though is that MYC has been reported as a downstream target of sonic hedgehog signaling, but not an upstream regulated before. So it suggests that actually sonic hedgehog and MYC may have maybe a bidirectional regulatory ability in terms of depending on whether it's being sent or received. And to ask the other, and so we can play a similar game, but we'll ask the other question. So given that a spot or cell has received sonic hedgehog signaling, what are the downstream transcription factors that most respond to this received signaling? And we do something different in this case, which is we take, again, the top module transcription factors, and instead of fitting a random forest model, because we have this continuous information, we can fit what are called generalized additive models to fit basically the expression of these transcription factors as a function of sonic hedgehog inflow, received sonic hedgehog signaling. If you've ever done anything like pseudotime analysis, it's basically a simplified version of that to understand which genes correspond, instead of which genes correspond or drive along a lineage, which genes are responding to an increase or receiving of a certain signal. And so in this case, we can plot, and in this case, we just fit the transcription factors as functions of sonic hedgehog inflow, and then we rank them by Spearman correlation. In this case, I've taken out the middle just so I could blow up the genes. There's many more you can see in the paper. And actually you can show that like you get quite a number of genes that are significantly negatively correlated, so which in that case indicates they may be downregulated by receiving sonic hedgehog signaling. And there are also quite a number that are positively correlated. And you can see here that it looks like they are increasing in expression based on receiving sonic hedgehog. And again, what's nice is that quite a few of these have been confirmed in the literature as like known targets of sonic hedgehog signaling. So it does tell us that at least FlowSig is finding the right structures in some sense, at least. I'm going to focus on one particular, and what we notice about when we're looking at these upstream and downstream regulators is that, for example, in this case, in the downstream targets, there was one transcription factor, FOXA2, which is positively correlated with receiving sonic hedgehog inflow. But at the same time, when we were looking at the upstream transcription factors that regulated sonic hedgehog, it's also one of the top upstream in terms of most predictive of sonic hedgehog outflow. So what this says is that both FOXA2 is a target of downstream target of received sonic hedgehog signaling, but which in itself also then drives an outflow of sonic hedgehog, which are two distinct measurements, I should say. But the point is here is that through this one transcription factor, it may be that sonic hedgehog is able to self-regulate itself, and in particular self-activate itself, given this positive correlation. And so this is something that we could find using this downstream analysis of the FlowSig network. We can go a step, to kind of build on this notion of co-regulation, we can go a step further. When we were looking at the networks of the upstream and downstream drivers of sonic hedgehog, we noticed that there were actually, what was interesting about these networks is that there were several ligands that seem to be both simultaneously upstream of sonic hedgehog outflow and downstream. So in this case, there are 5 ligands, BMP4, CXCR12, IGF2, MDK, Midkine, and WID5A that seem to be both upstream and downstream of sonic hedgehog. So the question we had to ourselves was, well, given that we're seeing this, is FlowSig suggesting, or the network suggesting, that there might be some sort of co-regulation or bidirectional regulation between Sonic Hedgehog and these other signals. And so to kind of try and figure this out, we use FlowSig to ask like two important downstream analysis questions. One, which is like for bidirectional regulation to be true of these ligands, they need to have two criteria be satisfied. One is that, for example, we're talking about BMP4, the downstream targets of BMP4 need to also be upstream transcription factor regulators of Sonic Hedgehog outflow. And then two, the converse also has to be true. The transcription factors that are upstream of, say, BMP4 need to be downstream targets of Sonic Hedgehog. And so we're kind of trying to find these loops by using the same analysis that we did before. And for any of these possible ligands, which of these is most likely to show some sort of bidirectional regulation? And so the first sanity check we did was to, well, ask ourselves which of these ligands of these five are even spatially possibly co-expressed with Sonic Hedgehog. So in this case, this is why I don't believe FlowSig is necessarily causal. It's more of an exploratory tool because not all of these ligands actually showed significant co-expression with Sonic Hedgehog. In fact, of those, the most significant, there were a couple, but the most significant signal that was like co-expressed with inflow and outflow was Win5a. In this case, what's important is that the inflow of Sonic Hedgehog seems to be co-expressed with the outflow of Win5a. So in that case, it may be that receiving Sonic Hedgehog drives you to express Win5a. In converse, you also are hoping that the receiving Win5a may drive an outflow of Sonic Hedgehog. And at least as a sanity check, looking at the spatial expressions, Win5a looked like a good candidate to start digging into possible co-regulation with Sonic Hedgehog, just based on spatial expression alone. But we can go a little further and basically play the same game as we did for looking for upstream and downstream regulators of Sonic Hedgehog, which is to go, okay, looking at these upstream drivers of Sonic Hedgehog, we can see that Win5a regulates Sonic Hedgehog through three intracellular modules, which have certain sets of transcription factors enriched for them. When we then do the same game with like, okay, we're looking for downstream, in this case, we're looking at downstream targets of Win5a, and we can fit generalized additive models, we can look at which are most significantly correlated. And one of the ones that stuck out to us was the fact that MYC, this transcription factor, is significantly negatively correlated with the received Win5a. So Win5a inflow. If you're receiving Win5a signaling, you tend to be downregulating MYC expression. But the interesting thing about that is the fact that, well, when MYC, as you may remember, is actually one of the more significant upstream regulators of Sonic Hedgehog outflow, which suggests to us that maybe MYC is both acting upstream of Sonic Hedgehog and downstream of Win5a. So you're getting this one channel where Win5a, receiving Win5a, is processed through the inhibition of MYC, which activates Sonic Hedgehog. So there's one regulatory pathway from Win5a, MYC to Sonic Hedgehog. In converse, we wanted to look at which downstream target of Sonic Hedgehog might be an upstream regulator of Win5a. And so here the key thing in the network is to realize only one module is mediating Sonic Hedgehog inflow, the Win5a outflow. It's module 5. So we can take the top transcription factors of this module and again fit a random forest model to ask which of these are significant predictors of Win5a inflow. And here there are three that stuck out for us, possibly four even: FOXA2, NK6X1, and SOX21. These were the three most significantly predictive transcription factors of Win5a outflow. And again, we realize when you look at we're kind of casting our minds back to our analysis of Sonic Hedgehog, these three transcription factors were also downstream targets of Sonic Hedgehog inflow. So the idea here is that in that case, your Sonic Hedgehog is being sent, it's being processed downstream by FOXA2, NKX61, and SOX21, which in then turn may actually regulate the outflow of Win5a. So you have another pathway from Sonic Hedgehog to Win via these three transcription factors. And so to put it all together, what this suggests is what we're calling an activator inhibitor modules. If you've ever studied Turing patterns in developmental biology, they're a very famous mathematical model proposed by Alan Turing as sort of the first way to, sort of the simplest model that generates spatially complex patterns that underlie morphogenesis and development in general. And it's been like this quest to figure out if activated inhibitor modules really do exist in nature, as based on Turing's models. And so in this case, we believe FlowSig suggested that such a module could exist between Sonic Hedgehog and Wnt5a. And it's based on the idea that, first of all, you have Sonic Hedgehog gene expression, which gets diffused and binds to then derive inflow of Sonic Hedgehog in nearby or in the same locations. In turn, receiving Sonic Hedgehog causes a self-activation via FOXA2, and also in turn activates Wnt5a outflow via these three transcription factors. In turn, that Wnt5a expression will then diffuse and bind to nearby spots and that inflow of that signal Wnt5a will actually then inhibit MYC expression, which we've shown drives an outflow of Sonic Hedgehog. And so you get the self-activating Sonic Hedgehog, which activates Wnt5a, which then in turn inhibits Sonic Hedgehog signaling. And so you have this like very nice activated inhibitor module that may be responsible for generating some of the more complex spatial patterns that you see in embryogenesis, or that's the hope. So in the interest of time, I'll summarize by saying we developed this method of FlowSig based on if you take cell-cell communication and gene expression data, you can infer these networks that describe if I'm receiving a signal, what signals am I likely to then send and through which intracellular mechanisms in a very coarse-grained manner. You can extract these very nice directed intercellular flow networks that describe upstream and downstream regulatory mechanisms of these different signals. And by teasing out the different, using gene expression values and using these networks to do certain downstream analysis, you can really tease out the specific like regulatory genes that are driving these flows, which allow you to then discover more complex regulatory motifs. This is just like, so FlowSig for us in many ways was a proof of concept, given that we have these descriptive analysis that are produced by cell-cell communication inference. Could we go a step further? Could we say something about their dependence relations? And could we say something about whether things are much more, which interactions are actually like significantly related or causally related to each other? The answer is like, we can't fully say whether things are causal for a number of reasons. And we would like to make FlowSig or expand on FlowSig to make it closer to saying something predictive. Right now, I would call it like an exploratory tool that forms the basis of where you should like start to kind of frame your analysis in terms of looking at which signaling affects which other signaling. To really kind of tease out the predictive power, though, we would like to embed some sort of notion of time in these models. The network kind of gives the impression that these things happen almost instantaneously. It's not really true. Signals take time to diffuse, they take time to then bind and activate transcription responses, and then which then take time to result in translation of proteins. There are inherent timescales of all these processes, and right now FlowSig doesn't account for that. Second is also based on not only making it predictive, but also making FlowSig more accurate. And the key thing about this is the fact that we're only using transcriptomic data to infer the intercellular flows. So in fact, just mRNA. But the reality is, though, cell-cell communication is really involves a number of processes. There's signaling of proteins that then activated transcription downstream response. And these transcription responses are regulated through effectively chromatin accessibility or like different enhancer promoter regions. And these different scales of information are better measured by, say, ATAC sequencing, proteomics, phosphoproteomics. And so we would like FlowSig to be able to incorporate these different data inputs. And then finally, based on like what I know people in this group study, I've only really talked about protein signaling and particularly like spatial diffuse secreted signaling, but there's also other types of things that facilitate cell-cell communication, metabolites, hormones, endocrine signaling in general, that we're really like, it is worth thinking about how do we kind of account for these processes as well. So I'll finally end, and sorry, I've taken a bit of time. I'll end by saying thank you to people in my lab, past and current colleagues that have really helped spawn the discussions that advance this work, Ching for really pushing this project along. And our collaborators in the Watanabe lab, Omoko Watanabe's lab, Wen Chen, and Momoko, who generously generated some of the organoid and validation data that allowed us to see that FlowSig was sort of heading in the right direction. Um, and I'll finish by just saying that the paper's out at Nature Methods. If you want to read more about it, I only presented like one major result really. And it's also available as a Python package that you can install. And yeah, I would love to get feedback and any questions. So thank you for listening to this talk.
Thank you all for this wonderful talk. Any questions, Sunil? I think you have something to say, I guess.
Yeah, sure. I mean, this is fascinating. Okay, I'm going to ask you a question that doesn't undermine anything that you have done. Okay, really good. My question basically is, of the numbers of signals that you identified in the embryo at the 9.5 stage, how many of these follow the protocol that you outline? So you know what I'm saying? So that for the RNAs, there were at least 150 signal dots that were there.
Yeah.
When you're doing all the bootstrapping and inferring, at the end of the day, how many of those 150 dots or signals do actually follow this, or are there multiple alternative pathways that you have yet to decipher?
That is a really nice question. And the answer, it's a bit of both, is the short answer. The longer answer is, yeah, if you have, say, like 150 of these interactions, what I've found is that maybe about like 10 or 20% of these seem to have some sort of dependencies. So a lot of the times it really is like it either seems to be a coincidence that they look like they're kind of being co-expressed, or it might just be that actually we haven't captured the right information to describe what is the dependency structures. It might, for example, like it's kind of like there's low-key knowledge that transcription factors aren't best measured by transcriptomic data. They're better measured by like chromatin accessibility measurements, right? So it might be that we failed to capture the right gene regulatory mechanism in that way. And then second, I think about these processes. And so that's, yeah, it's effectively a fraction. And then, sorry, what was the second part of the question?
So that's the one part. The second part, of course, is there tissue dependency? That do you? And I didn't really ask you that question, but that was the question that follow-up that I was waiting to ask once you answered.
No, that's again an excellent question. And so in this case, like for this spatial data at the time, the module we were using, the method we were using, non-negative spatial factorization, was only for one tissue sample. But actually in this data, there are multiple samples at E9.5. So what you would arguably do is you would say infer a network on one sample and then test your predictions on another sample. Or you could use multiple samples to infer, like, have more confidence in the statistical relations you're inferring. There are now, that method, NSF has been extended to now incorporate multiple samples, so you could do that. But I think actually what would be more useful, and I didn't think of it at the time, is you would learn these dependent relations in one sample and then test on another cross-section or something like that.
Well, no, you're going a slightly wider than I was thinking of.
That's okay.
What I was thinking of is, if you find a relationship in the brain area, would that work also for the tail area, or in the liver, the lung, the heart, or whatever? Are those relationships constants, or does the tissue of origin, so to say, modify these reactions in variety of sorts? And if so, how do you think about those? So
yes, I get what you mean now. So the short answer is that's why we introduce those intracellular modules. So the idea of those modules is they capture information about cell type specificity. So they can capture information about cell state enrichment in different spots, and also about, say, spatial, like, locality. So the idea is that these modules are locally expressed rather than, say, being globally expressed most of the time. And so in that, the assumption I had is like, if you see Sonic Hedgehog infecting Win5a, that relation doesn't necessarily hold for the entire tissue. It holds in very specific parts depending on specific regulatory mechanisms that are specific to like a location or a cell state. So the idea is like we're trying to capture that specificity or niche dependent relation through the intracellular module expressions in a very coarse-grained manner. You could what you would really hope to do is actually try and tease out the specific gene regulatory networks. But if you do that, you're introducing an exponentially greater number of nodes, which then makes it computationally intractable and a little harder to like do the inference on, so to speak. So yeah, the modules are meant to take care of that specificity, so to speak.
Anyway, again, phenomenal talk. Good work.
Thank you.
Great work.
So, well, there's one question in the chat, and it is basically if the ligand signaling flow and the downstream genes secreted are present exactly at the same spot or spatial location at the same time, how do we distinguish between the flow of communication if prior knowledge of ligand and cellular process downstream gene is not utilized? So that's also on the chat. You can read as well.
So in that case, if prior knowledge wasn't utilized, so this is it's a good question in that a lot of this method was based around what happens if we don't use downstream networks. So I should say actually like for non-spatial inference, we do use some sort of prior knowledge, which is that to infer the amount of received signal, you need to know some information about if I have a certain ligand-receptor interaction, what are the downstream transcription factors? Because unlike with spatial data, for non-spatial data, you cannot infer from which signal you're receiving. Like you cannot infer the amount of Wnt5a signal. You can only infer, like say, whether an interaction has there is sufficient downstream activation. So in that case, we use the prior knowledge to construct signal inflow variables. In this case, you could, if you wanted to use prior knowledge to infer, say, ligands and then downstream transcription factors, you very well could. There's two ways of going about this in the field. One is that you can embed this directly into the network by introducing prior knowledge. So you can say actually like, given that certain modules express transcription factors, which are known to be downstream of certain ligands, you can then kind of start your graph search on at certain edges to incorporate that prior knowledge. The other way is to effectively use it as a sort of pseudo validation at the end. You know that if you know that certain transcription factors are downstream of certain ligands, that tells you actually, it gives you a way of ruling out false positives, or it also gives you a starting point to then perform like the generalized additive model analysis to work out what are the likely downstream responses. So there's a bit of a roundabout answer of myself, but effectively you can incorporate it directly into the graph, or you can kind of use it as to filter out some of the module-specific transcription factors when looking at downstream responses. Yeah.
That's all. Thank you. Are there any questions? Please feel free to unmute or post on the chat. So in the meantime, so the cell communication, right, ligand receptors, you're looking at this prior information, but physically, how are they interacting, right? And what's the exactly in touch and what's not exactly in touch? Can this be incorporated into models? I'm curious about, there's this AlphaFold looking at all these protein pairs, right? Could you help us understand where we are with this?
That is a really good question. And that is the thing that like, as someone who came from like a mathematical modeling background, when I looked at biology, it was often through this sort of biophysical mechanistic lens of like signals diffuse and then bind and trigger some sort of interaction. And one of the challenges is that a lot of that is lost with like looking at like gene expression data. There are new technologies that are a little better at it, but like it is a really good question. And the simplest answer, the answer I can give right now is that it is sort of incorporated through the optimal transport algorithms that my colleague Jishen described, which is like, even though technically optimal transport is devoid of mechanism, it does a very good job of accounting for the fact that these signals need to diffuse in space. But in terms of actual physical binding, I think this is something where, in that sense, like that's kind of the limit of using gene expression to understand cell-cell communication, which really requires you to have some sort of protein information. And in that case, you could use AlphaFold to look at binding, or you could even, I think something I'm really interested in is how do you use something like, say, phosphoproteomics to understand where are the actual post-translational modifications and where are the actual like inflows of signaling occurring. So in that sense, yeah, I've not thought about how best to use AlphaFold in that sense. I should, considering it just won a Nobel Prize. But effectively, my instincts are to kind of broaden the data input to not just include transcriptomic, but to also include protein scale information or other sort of modalities. Yeah.
Yeah, but
Wonderful.
as you start thinking about it, there's so many additional parameters. Classically, it will be splicing.
Yes. Yes.
That would create a lot of havoc as we start thinking about interactions, because both RNA splicing, protein, which would lead to alternative protein structures, could have completely different dramatic impact on what happens in terms of cell-cell interactions and ligand protein interactions. So
I'm so glad you brought that up, because I've been wondering this as well. Like, what is something that's been in the back of my head is like, but now with long read sequencing, we're getting much better understanding transcript isoforms, right? And we can even infer like demultiplex like a nascent and mature mRNA these days. And so I've been wondering like, what is actually the effect of that on cell-cell communication? And like, yeah, what is the effect of like different isoforms, alternative splicing, and say the dysregulation of that on, yeah, affecting like what signals are effectively sent and what are received, right? Yeah, no, I love that.
And furthermore, it seems like there's, whenever we talk about alternative splicing, we think that it's form A or form B or form C. One of the three options are there, but it seems that what changes is the frequency of different forms rather than the absolute form. Everything from, instead of form A being form B, it's not that, it's the frequency changes.
Yeah.
So with that, you get a wide array of different splice isoforms, which would result in a wide array of different proteins, which would further interact all the interactions and the interactome that would be associated with it. So unfortunately, life is complicated.
Absolutely. But you do touch on something that I think is very important, that like for myself, or at least with FlowSig, like right now it's very descriptive, it's very static. There's not a lot of quantitative analysis in the sense of like, it doesn't give you distributions, or it doesn't give you like, what is the effect of X and Y? And so I think thinking really hard about like, how do we model this sort of signals in a probabilistic sense, I think is like a really important step forward to actually like doing something quantitative or predictive with these methods. So yeah, I like this. Yeah, this frequency of isoforms is a very nice point. Yeah, thank you for bringing it up.
Yeah, but you take a very important first step in that direction. So I really like your work.
Thank you.
Wonderful. A lot of exciting ideas, but I think in the interest of time, we'll wrap up today. Let's thank Axel for this wonderful talk. I'll be using this clapping sign here too. Thank you so much for this wonderful talk, and appreciated your time and insights onto cell communication. And also the follow-up discussion was really helpful. Thank you, Dr. Badra, for this. So we will wrap up right now, but before we conclude, I'd like to highlight that in 2 weeks from now, we will have Dr. Daiwei Zhang from University of North Carolina, Chapel Hill, and he's going to talk about inferring super-resolution tissue architecture by integrating spatial transcriptomics with histology. I think this was a Nature Biotech paper, as far as I remember. So please tune in 2 weeks, and thank you again, everyone, for participating today, and hope to see you guys in 2 weeks. Have a nice weekend.
Thank you again for the invitation.
Thank you, Axel. Have a nice one.